1 General Introduction. Introduction. Medical parasitology deals with the parasites, which cause human infections and the diseases they produce. It is broadly divided into 2 parts. 1. Protozoology. 2. Helminthology. The pioneer Dutch microscopist, Antonie Leuven and Leeuwenhoek of Holland in 1681, first introduced single-lens microscope and observed Giardia in his own stools. Louis Pasteur in 1870, first published scientific study on a protozoa, disease leading to its control and prevention during investigation of an epidemic silkworm disease in South Europe. A seminal discovery was made in 1878 by Patrick Manson about the role of mosquitoes in filariasis. This was the first evidence of vector transmission. Afterwards, Laveran in Algeria discovered the malarial parasite, 1880, and Ronald Ross in Secunderabad and Calcutta in India showed its transmission by mosquitoes, 1897. A large number of vector-borne diseases have since then been identified. By mid-20th century, with dramatic advances in antibiotics and chemotherapy, insecticides and antiparasitic drugs, and improved lifestyles, all infectious diseases seemed amenable to control. Parasites are living organisms, which depend on a living host for their nourishment and survival. They malreplicate or undergo development in the host. The term parasite is usually applied to protozoa, unicellular organisms, and helminths, multicellular organisms. Flowchart 1. Parasites can also be classified as ectoparasite. Ectoparasites inhabit only the body surface of the host without penetrating the tissue. Lice, ticks and mites are examples of ectoparasites. LHE parasitology term infestation is often employed for parasitization with ectoparasites. Endoparasite, a parasite, which lives within the body of the host and is said to cause an infection, is called an endoparasite. Most of the protozoan and helminthic parasites causing human disease are endoparasites. Free living parasite. It refers to non-parasitic stages of active existence, which live independent of the host. For example, cystic stage of Naegleria fowleri. Endoparasites can further be classified as obligate parasite, the parasite, which cannot exist without a host. For example, Toxoplasma gondii and Plasmodium facultative parasite, organism which may either live as parasitic form or as free living form. For example, Naegleria fowleri. Accidental parasites, parasites, which infect an unusual host are known as accidental parasites. Echinococcus granulosus infects man accidentally, giving rise to hydatid cysts. Aberrant parasites, parasites, which infect a host where they cannot develop further are known as aberrant or wandering parasites. For example, Toxocara canis, dog roundworm, infecting Leeuwenhoek. Host is defined as an organism which harbors the parasite and provides nourishment and shelter to latter and is relatively larger than the parasite. The host may be of the following types. Definitive host, the host in which the adult parasite lives and undergoes sexual reproduction is called the definitive host. For example, mosquito acts as definitive host in malaria. The definitive host may be a human or any other living being. Accidental parasites, parasites, which infect an unusual host are known as accidental parasites. Echinococcus granulosus infects man accidentally, giving rise to hydatid cysts. Aberrant parasites, parasites, which infect a host where they cannot develop further are known as aberrant or wandering parasites. For example, Toxocara canis, dog roundworm, infecting Leeuwenhoek. Host is defined as an organism which harbors the parasite and provides nourishment and shelter to latter and is relatively larger than the parasite. The host may be of the following types. Definitive host, the host in which the adult parasite lives and undergoes sexual reproduction is called the definitive host. For example, mosquito acts as definitive host in malaria. The definitive host may be a human or any other living being. However, in majority of human parasitic infections, man is the definitive host. For example, filaria, roundworm, hookworm. Panica's Textbook of Medical Parasitology. Flowchart 1. Types of parasites. Parasite plus protozoa. Unicellular. Kingdom Protista plus helminths. Multicellular. Kingdom. Animalia Amabi flagellates. Sporozoaciliates plus. Enamoeba Neglina Giardia Trichomonas Plasmodium Babesia Toxoplasma Balanidium nematodes. Ascaris ancylostoma. Intermediate host. The host, in which the larval stage of the parasite lives or asexual multiplication takes place, is called the intermediate host. In some parasites, 2 different intermediate hosts may be required to complete different larval stages. These are known as first and second intermediate hosts, respectively. Box 1. Paratenic host. A host, in which larval stage of the parasite remains viable without further development, is referred as a paratenic host. Such host transmits the infection to another host. For example, fish for Pleurocercoid larva of D. lalum. Reservoir host. In an endemic area, a parasitic infection is continuously kept up by the presence of a host, which harbors the parasite and acts as an important source of infection to other susceptible hosts. For example, dog is the reservoir host of hydatid disease. Accidental host, the host, in which the parasite is not usually found. For example, man is an accidental host for cystic echinococcus. Zoonosis. The word zoonosis was introduced by Rudolf Virchow in 1880 to include the diseases shared in nature by man and animals. Later, in 1959, the World Health Organization, WHO, defined zoonosis as those diseases and infections which are naturally transmitted between vertebrate animals and man. Cestodes, Taenia echinococcus trematodes plus Fasciola schistosoma. Box 1. Parasites with man as intermediate or secondary host. Plasmodium spp, Babesia spp, Toxoplasma gondii, Echinococcus granulosus, Echinococcus multiloco aeris, Taenia solium, Spirometer spp. It is of following types. Protozoal zoonoses, for example Toxoplasmosis, Leishmaniasis, Balangladiosis and Cryptosporidiosis. Ileominthic zoonosis, for example hydatid disease, Tinniasis. Anthropozoonoses, infections transmitted to man from lower vertebrate animals, for example cystic echinococcus. Zoanthroponosis, infections transmitted from man to lower vertebrate animals, for example human N1 tuberculosis to cattle. Host-parasite relationships are of following types. Flowchart 2. Symbiosis, commensalism, parasitism. General Introduction. Parasitology. Flowchart 2. Host-parasite relationships. Host. Parasite relationships are 1. Symbiosis, commensalism, parasitism. Both host and parasite are dependent upon each other. None of them suffers any harm from the association. Only the parasite derives benefit from the association without causing any injury to the host. The parasite derives benefits and the host is always harmed due to the association. A commensal is capable of living an independent life also. The parasite cannot HVE an independent life. Life cycle of parasites. Direct life cycle. When a parasite requires only single host to complete its development, it is called as direct life cycle. For example, Entamoeba histolytica requires only a human host to complete its life cycle. Table 1. Indirect life cycle. When a parasite requires 2 or more species of host to complete its development, the life cycle is called as indirect life cycle. For example, malarial parasite requires both human host and mosquito to complete its life cycle. Tables 2 and 3. Common sources of infection: contaminated soil and water. Soil polluted with embryonated eggs. Roundworm, whipworm, may be ingested, or infected larvae in soil, may penetrate exposed skin. Hookworm. Dartlenfeclive forms of parasites present in water may be ingested. Cyst of amoeba and giardia. Water containing the intermediate host may be swallowed. Cyclops containing guinea worm larva. Infected larvae in water may enter by penetrating exposed skin. Cercaria of Schistosom's free-living parasites in water may directly enter through vulnerable sites. Naegleria may enter through nasopharynx. Food. Ingestion of contaminated food or vegetables can reigning infeclive stage of parasite. Amoebic cysts. Toxoplasma oocysts. Echinococcus eggs. Ingestion of raw or undercooked meat harboring infeclive larvae larvae, measly port-containing Cysticercus cellulosae, the larval stage of Taenia solium. Vectors. A vector is an agent, usually an arthropod that transmits an infection from man to man or from other animals to man. For example, female Anopheles is the vector of malarial parasite. Vectors can be biological vectors. The term biological vector refers to a vector, which not only assists in the transfer of Table 1, parasites having direct life cycle, requiring no intermediate host. Protozoa helminths, Entamoeba histolytica, Ascaris lumbricades, Giardia lamphia, Enterobius vermicularis, Trichomonas vaginaris, Trichuris trichiura, Balantidium coli, Ancylostoma duodenale, Cryptosporidium pavum, Necator americanus, Comocyclospora carinensis, Hymenolopus nana, Sospora B2, Microsporidia parasites. But the parasites undergo development or multiplical line in their body as well. They are also called as true vectors. Example of true vectors are mosquito, malaria, filariasis, sandflies, Cholera, tsetse flies, sleeping sickness, reduvial bugs, Chagas disease, ticks, babesiosis. Mechanical vectors. The term mechanical vector refers to a vector which assists in the transfer of parasitic form between hosts but is not essential in the life cycle of the parasite. Example of mechanical vectors is housefly, amoebiasis. In biological vectors, a certain period has to elapse after the parasite enters the vector before it becomes infective. This is necessary because the vector can transmit the infect line only after the parasite multiplies to a certain level or undergoes a developmental process in its body. This interval between the entry of the parasite into the vector and the time it takes to become capable of transmitting the infection is called the extrinsic incubation period. Animals, domestic, cow, for example T. saginata, Sarcocystis. Panica's Textbook of Medical Parasitology Table 2. Parasites having indirect life cycle requiring one intermediate. Box 2. Parasites causing autoinfection host and one definitive host. Parasite definitive host intermediate host. Protozoa Plasmodium spp. Female Anopheles man mosquito babesia tick man leishmania man dog sandfly trypanosoma brucei man tsetse fly trypanosoma cruzi man triatamine bug toxoplasma gondii cat man cestodes Taenia solium man pig Taenia saginera man catalichinococcus. Granulosus dog man trematodes fascia hepatica man snail fascia apsis busky man pig snail schistosoma spp. Man snail nematodes trichinella spiralis man pig watchereria bancrofti man mosquito brugia malayi man mosquito dracunculus medinensis man cyclops. Table 3. Parasites having indirect life cycle requiring two intermediate host and one definitive host. Parasite intermediate hosts definitive host fascia spp. Snail, plant man Clonorchis sinensis snail, fishman Diphyllobothrium latum cyclops, fishman Paragonimus wistemani snail, crustacean man, pig, for example T. solium, Trichinella spiralis, dog, for example Echinococcus granulosus, cat, for example Toxoplasma, Opus faucus, wild game animals, for example Trypanosomiasis, wild felines, for example Paragonimus wistemani, fish, for example fish tapeworm, mollusks, for example liver flukes, copods, for example guinea worm. Carrier, a person who is infected with parasite without any clinical or subclinical disease is known as carrier. He can transmit parasite to others, for example, all, Hymenolopus nana, Enterobius vermicularis, Taenia solium, Strongyloides stercoralis, Capillaria philipinensis, Cryptosporidium pavum and trypanotic infections. Vertical transmission of congenital infections, self, auto-infection. Box 2, finger-to-mouth transmission, for example pinworm internal reinfection, for example strongyloides. Modes of infection oral transmission. The most common method of transmission is through oral route by contaminated food, water, soiled fingers, or fomites. Many intestinal parasites enter the body in this manner, the infective stages being cysts, embryonated eggs, or larval forms. Infection with E. histolytica and other intestinal protozoa occurs when the infective cysts are swallowed. Skin transmission. Entry through skin is another important mode of transmission. Hookworm infection is acquired when the larvae enter the skin of persons walking barefooted on contaminated soil. Schistosomiasis is acquired when the Cercarial larvae in water penetrate the skin. Vector transmission. Many parasitic diseases are transmitted by insect bite. For example, malaria is transmitted by bite of female Anopheles mosquito. Filariasis is transmitted by bite of Culex mosquito. A vector could be a biological vector or a mechanical vector. Direct transmission. Parasitic infection may be transmitted by person-to-person contact in some cases, for example by kissing in the case of gingival and bisexual intercourse in trichomoniasis. Vertical transmission. Mother-to-fetus transmission may take place in malaria and toxoplasmosis. Iatrogenic transmission. It is seen in case of transfusion malaria and toxoplasmosis after organ transplantation. Pathogenesis. Parasitic infections may remain inapparent or give rise to clinical disease. A few organisms, such as E. histolytica, may live as surface commensals, without invading the tissue. Clinical infection produced by parasite may take many forms: acute, subacute, chronic, latent, or recurrent. Pathogenic mechanisms, which can occur in parasitic infections are lytic necrosis. Enzymes produced by some parasite can cause lytic necrosis. E. histolytica lyses intestinal cells and produces amoebic ulcers. Trauma. Attachment of hookworms on jejunal mucosa leads to traumatic damage of villi and bleeding at the site of attachment. Allergic manifestations. Clinical illness may be caused by host immune response to parasitic infection. For example, eosinophilic pneumonia in Ascaris infection and anaphylactic shock in rupture of hydatid cyst. Physical obstruction. Masses of roundworm cause intestinal obstruction. Plasmodium falciparum malaria may produce blockage of brain capillaries in cerebral malaria. Inflammatory reaction. Clinical illness may be caused by inflammatory changes and consequent fibrosis. For example, lymphadenitis in filariasis and urinary bladder granuloma in schistosoma hemolobium infection. Neoplasia. A few parasitic infection have been shown to lead to malignancy. The liver fluke. Clonorchis may induce bile duct carcinoma, and S. hematobium may cause urinary bladder cancer. Space-occupying lesions. Some parasites produce cystic lesion that may compress the surrounding tissue or organ, for example hydatid cyst. Immunity in parasitic infection. Like other infectious agents, parasites also elicit immunoresponses in the host, both humoral as well as cellular. Figure 1. But immunological protection against parasitic infections is much less efficient than it is against bacterial or viral infections. Several factors may contribute to this. Compared to bacteria and viruses, parasites are enormously larger or more complex structurally and antigenically, so that the immune system may not be able to focus attack on the protective androgens. Many protozoan parasites are intracellular in location, and this protects them from immunological attack. Several protozoa and helminths live inside body cavities. 1H's location limits the efficiency of immunological attack. Once the parasitic infection is completely eliminated, the host becomes again susceptible to reinfection. This type of immunity to reinfection is dependent on the continued presence of residual parasite population and is known as premunition. Antibodies belonging to different immunoglobulin classes are produced in response to parasitic infections. Selective tests for immunoglobulin M, IgM, are helpful in differentiating current infections from old infections. Excessive IgE response occurs in helminthiasis. A characteristic cellular response in helminth parasite is eosinophilia, both local and systemic. Figure 1. Parasites have evolved to be closely adapted to the host, and most parasitic infections are chronic and show a degree of host specificity. For example, malarial parasites. General introduction. Parasitology. Figure 1. Eosinophils surrounding schistosomulum, an example of immune attack in bloodstream. Box 3. Parasites exhibiting antigenic variations. Trypanosoma brucei gombeans, Trypanosoma brucei rhodesians, Plasmodium spp, Giardia Giardia lamblia of human. Bird and rodents are confined to their own particular species. Parasites like trypanosomes exhibit antigenic variation within the host. This genetic switch protects them from antibodies. Similar mechanism may be operative in the recrudescences in human malaria. Box 3. Some parasites adopt antigenic disguise. Their surface antigens are so closely similar to host components that they are not recognized as foreign by the immune system. Some infections may produce immunodeficiency due to extensive damage to the reticuloendothelial system, as in case of visceral leishmaniasis. The fact that immunity normally plays an important role in the containment of parasitic infections is illustrated by the florid manifestations caused by opportunistic parasites such as Pneumocystis jirovecii and T. gondii. When the immune response is inadequate as in acquired immunodeficiency syndrome, AIDS, and other immunodeficiencies, immune evasion, all animal pathogens, including parasitic protozoa and worms, have evolved effective mechanism to avoid elimination by the host defense system as described in Table 4. Vaccination. No effective vaccine for humans has so far been developed against parasites due to their complex life cycles, adaptive responses and antigenic variation. Great progress has been. Panica's Textbook of Medical Parasitology. Table 4. Parasite escape mechanisms. Parasite escape mechanisms. Example: intracellular habitat. Malarial parasite. Leishmania. Encystment. Toxoplasma. Trypanosoma cruzi. Resistance to microbial phagocytosis. Leishmania. Masking of antigens. Schistosomes. Variation of antigen. Trypanosomes. Plasmodium spp. Suppression of immune response. Trichinella spirosa. Schistosoma mansoni. Malarial parasite. Interference by polyclonal trypanosomes. Activation. Sharing of antigens between parasite schistosomes and host. Molecular mimicry. Continuous turnover and release of schistosomes. Surface antigens of parasite made in identifying protective antigens in malaria and some other infections, with a view to eventual development of prophylactic vaccines. Laboratory diagnosis. Most of the parasitic infection cannot be conclusively diagnosed on the basis of clinical features and physical examination. Laboratory diagnosis depends upon microscopy, culture, serological test, skin test, molecular method, animal inoculation, xenodiagnosis, imaging, hematology, microscopy. An appropriate clinical specimen should be collected for definitive diagnosis of parasitic infections. Following specimens are usually examined to establish a diagnosis: stool, blood, urine, sputum, cerebrospinal fluid, CSF, tissue and aspirates. Genital specimens. Stool examination. Examination of stool is very important for the detection of intestinal infections like Giardia, Enlomoeba, Ascaris, Ancylostoma, etc. Cysts and lymphocytes of E. histolytica. C. lamblia can be demonstrated in fesses. Eggs of roundworm and tapeworm are also found in stool. The larvae are found in the fesses in S. slow-coriolis infection. Table 5. For further details, refer to Chapter 23. Blood examination. Examination of blood is of vital importance for demonstrating parasites which circulate in blood vessels. Table 6. Malarial parasite is confirmed by demonstration of its morphological stages in the blood. Urine examination. The characteristic lateral spined eggs of S. hematobium and tryphozoites of T. vaginaris can be detected in urine. Microfilaria of W. bancrofti are often demonstrated in the chylous urine. Box 4. Sputum examination. LHE eggs of P. wistermani are commonly demonstrated in the sputum specimen. Occasionally, larvae stages of S. coriolis and A. lumbricoides may also be found in sputum. Cerebrospinal fluid examination. Some protozoa like T. brucei, Nigleria, Acanthamoeba, Balamalhia and Angiostrongylus can be demonstrated in the CSF. Tissue and aspirates examination. The larvae of Trichinella and eggs of Schistosoma can be demonstrated in the muscle biopsy specimens. By histopathological examination of brain, Nigleria and Acanthamoeba can be detected. In Kyla Leishman Donovan, Low. Bodies can be demonstrated in spleen and bone marrow aspirate. Tryphozoites of Giardia can be demonstrated in intestinal aspirates. Tryphozoites of E. histolytica can be detected in liver pus in cases of amoebic liver abscess. Genital specimen examination. Tryphozoites of T. vaginalis are found in the vaginal and urethral discharge. Eggs of E. vermicularis are found in anal swabs. General introduction. Parasitology. Table 5. Parasites and their developmental stages found in stool cysts. Tryphozoites. Entamoeba histolytica. Giardia lamblia. Balantidium coli. Sarcocystis spp. Lysospora b2. Ceoclospora catanensis. Cryptosporidium parvomegs. Cestodes. Taenia spp. Hymenolopis nana. Hymenolopis diminuta. Oipidium caninum. Ophiloborium adam T. rematodes. Schistosoma spp. Fasciolopsis busci. Fasciora hepatica. Fasciora gigantica. Clonorchis sinensis. Table 6. Parasites found in peripheral blood film protozoa. Plasmodium spp. Babesia spp. Trypanosoma spp. Leishmania spp. Box 4. Parasites found in urine. Schistosoma hematobium. Muchereria bancrofti. Trichomonas vaginaris culture nematodes. Muchereria bancrofti. Brugia malayi. Loloa, Manzanella spp. Larvae adult worms. Gasserodiscoides hominis strongyloides stercoriolis. Taenia solium. Heterophyes. Metagonimus eucogoe. Opis thorchis spp. Nematodes. Trichuris trichiura. Enterobius vermicularis. Ascaris lumbricoides. Ancylostoma duodenale. Nicata americanus. T. richus rhongylus orientalis. Taenia saginata. Ophilobophoreum latum. Ascaris umbriculates. Enterobius vermicularis. Comma T. archine la spiro is Table 7. Antigen detection in parasitic diseases. Galactose lectin antigen. Giardia specific antigen 65. WKK and RK39 antigen. HRP. 2 antigen. Vivax specific PLDH. 200K diag and OG4C3 antigen. Entamoeba histolytica. Giardia lamblia Leishmania denovani Plasmodium falciparum Plasmodium vivax Muchereria bancrofti abbreviations. AGD. Antigen. HRP2. Histidine rich protein 2. PLDH. P. falciparum lactate dehydrogenase. RK39. Recombinant kinesin 39. WKK. Witkowski. Klingenstein and Kuhn by rapid immunochromatographic test. Filarial antigens are detected in current infection by enzyme-linked immunosorbent assay. ELISA. Table 7. Some parasites like Leishmania, Entamoeba and Trypanosoma can be cultured in LH laboratory in various azenic and polyxenic media. Antibody detection. The following antibody detection procedures are useful in detecting various parasitic infections like amoebiasis, echinococosis and leishmaniasis in man. Serological tests. Serological tests are helpful for the detection and surveillance of many protozoa and helminthic infections. These tests are basically of two types. 1. Tests for antigen detection. 2. Tests for antibody detection. Antigen detection. Malaria antigen like P. falciparum lactate dehydrogenase, PLDI, and histidine rich protein 2, HRP2, are detected. Complement fixation test, CFT, indirect hemagglutination EHA, indirect immunofluorescent antibody, IFA, test, rapid immunochromatographic test, IC, enzyme-linked immunosorbent assay test, ELISA. Skin tests. Skin tests are performed by injecting parasitic antigen intradermally and observing the reaction. In immediate hypersensitivity reaction, wheal and flare response is seen within 30 minutes of infection, whereas erythema and Panica's Textbook of Medical Parasitology. Box 5. Important skin tests done in parasitology. Cassini's test done in hydatid disease. Montenegro test or Leishmanin test done in Kyla. Azar. Frenkel's test done in toxoplasmosis. Fairley. S test done in schistosomiasis. Bachmann intradermal test done in trichinosis in duration seen after 48 hours of injection is called as delayed hypersensitivity reaction. Box 5. Molecular diagnosis. Molecular methods most frequently used to diagnose human parasitic infection are deoxyribonucleic acid, ANR, probes, polymerase chain reaction, PCR, and microarray technique. 1. These tests are very sensitive and specific. Animal inoculation. It is useful for the detection of toxoplasma, trypanosoma and babesia from the blood and other specimens. Xenodiagnosis. Some parasitic infection like Chagas disease caused by T. cruzi can be diagnosed by feeding the larvae of Reducus UVI bugs with patient's blood and then detection of mastocytes of T. cruzi in their fesses. Imaging procedures like X-ray, ultrasonography, USG, computed tomography, CT, scan and magnetic resonance imaging, MRI, are now being extensively used for diagnosing various parasitic infections like neurocysticercosis and hydatid cyst disease. Hematology anemia is frequently seen in hookworm infection and malaria. Eosinophilia is frequently present in helminthic infections. Hypogammaglobulinemia occurs in visceral leishmaniasis. Leukocytosis is seen in amebic liver abscess. Key points: Leeuwenhoek in 1681 first observed the parasite Giardia in stools. Laveran in 1880 discovered malarial parasite, and Ronald Ross in 1897 showed the transmission of malaria by mosquitoes. Protozoa belong to kingdom Protista and helminths belong to kingdom Animalia. Definitive host: the host in which the adult stage lives or the sexual mode of reproduction takes place. Intermediate host: the host in which the larval stage of the parasite lives or the asexual multiplication takes place. Zoonoses: diseases which can be transmitted to humans from animals, for example malaria, leishmaniasis, trypanosomiasis and echinococcus. Parasites like trypanosomes exhibit antigenic variation within the host. Parasites like Ascaris and echinococcus cause allergic manifestations in the host. Innate immunity against parasite may be genetic or by non-specific direct cell mediated or by complement activation. Acquired immunity in parasitic infections is by generating specific antibodies and effector T cells against parasitic antigens. Diagnosis of parasitic infections are made by direct identification of parasite in specimens like stool, blood, urine, bone marrow, CSF, sputum, etc. Serological tests are also useful in diagnosis by detection of parasite-specific antibody and antigen. Other diagnostic modalities include imaging, molecular methods like PCR, skin test and zoonodiagnosis. Review questions 1. Write short notes on A. Parasites, B. Host, C. Host parasite relationship, D. Zoonosis, E. Immune evasion mechanism of the parasites. 2. Discuss briefly the laboratory diagnosis of parasites. 3. Describe immunity in parasitic infections. 4. Differentiate between A. Direct and indirect life cycle, B. Definitive host and intermediate hosts. Multiple choice questions. 1. Definitive host is 1A in which sexual multiplication takes place and harbors adult form, B in which asexual multiplication takes place and harbors adult form, C. Other diagnostic modalities include imaging, molecular methods like PCR, skin test and zoonodiagnosis. Review questions 1. Write short notes on A. Parasites, B. Host, C. Host parasite relationship, D. Zoonosis, E. Immune evasion mechanism of the parasites. 2. Discuss briefly the laboratory diagnosis of parasites. 3. Describe immunity in parasitic infections. 4. Differentiate between A. Direct and indirect life cycle, B. Definitive host and intermediate hosts. Multiple choice questions 1. Definitive host is 1A in which sexual multiplication takes place and harbors adult form, B in which asexual multiplication takes place and harbors adult form, C in which sexual multiplication takes place and harbors larval form, D in which asexual multiplication takes place and harbors adult form. 2. Autoinfection is seen in all except A. Hemino-Epis nano B Enterabius vermicularis C. Tinea SOI UMD Ascaris lumbricoides. 3. Antigenic variation is exhibited by A. Entomoeba B. Schistosoma C. T. Rypanosoma D. Leishmania. 4. Which parasite enters the body by piercing the skin? A. Trichuris trichiura B. Ascaris C. Necator americanus D. Plasmodium. 5. Which parasitic infection leads to malignancy? A. Babesiosis B. Clonorchis sinensis C. Trypanosoma cruzi D. Schistosoma hematobium. 6. Zoonodiagnosis is useful in A. Wuchereria bancrofti B. Trypanosoma cruzi C. Trichinella spiralis D. All of the above. 7. The following is zoonotic disease except A. Leishmaniasis B. Balantidiasis C. Scabies D. Tinniasis. General introduction. Parasitology. 8. 2 hosts are required in A. Tinea solium B. Entamoeba histolytica C. Trichuris trichiura D. Giardia. 9. Which of the following parasite passes its life cycle through 3 hosts? A. Fascia hepatica B. Fascia robusci C. Schistosoma hematobium D. Clonorchis sinensis. 10. Man is the intermediate host for A. Strongyloides stercoralis B. Plasmodium vivax C. Entamoeba histolytica D. Enterobius vermicularis. Answer 1 or 2 D. 8 or 9 D. 3. C. 10 B. 4. C. 5 B. 6 D. 7. Capital C.